Good morning, biology students. Today is Wednesday, April 15th, and this is going to be your daily message. It's designed to work through what the expectations are for today's e-learning, as well as how to navigate some of the items on the class pages. So we're going to start here on the home page like we usually do. Birthday shout outs, I did not change it. I did not see any for spring break when we were out from 4/7 on. I will be updating those, so I'll give the shout outs as soon as I see one. If I did miss your birthday, please let me know. Send me an email. I went through Power School and I think I got them all, but just let me know. So, weekly agendas in detail. Let's go to week 14. That's where we're at. And when I click on that, this is what we see. Notice that you will see some differences from when you looked at it on Monday. And the reason is because I gave a protein synthesis screencast. You guys did that molecular biology self-paced module through Google Slides where you had to look at DNA, look at protein synthesis, watch a video, do an interactive. And so that was all self-paced. Today, what you're going to do is if you are still confused, you need a little bit more assistance, I did break up protein synthesis into two screencasts because it was kind of long. Feel free to watch these. These are optional, okay? If you feel like you have a good grasp on the slide, the self-paced slides that I gave you, if you feel you had a good grasp on doing this patient case study that you started on Monday, you might be able to just go on without watching the screencast. But otherwise, go ahead and go through it. I think I'm actually going to put the slides on them too. So if you just want to go through the slides without listening to me talking, I'm going to end up putting them over here. I'll probably put it right somewhere on the right column here. Okay, so feel free to use that. So if you're comfortable with protein synthesis so far and you don't need to do this part, then you're just going to go on to number 2. Otherwise, you're going to watch the screencast or go through the slides. In addition, we do have the protein synthesis with Amoeba Sisters. I posted it here. And if you're struggling to read a codon chart or a codon wheel, there is an Amoeba Sisters. I know I use these a lot, but it's because they break it down really simply with some good visuals for it. This video will help you to read a codon chart. And don't forget, I always have textbooks readings down here. So if you don't understand how to translate, if you don't understand the genetic code, the codon chart, go to the appropriate reading. And that's basically your textbook. Last thing I have here is I have a Protein Science Wrap Academy. Basically, it's going to review what a protein is, and it talks a little bit about protein synthesis. And I like those dorky science music videos, and so that's what this one is right here that I found, Science with Tom. So once you feel like you have a better grasp on protein synthesis, or if you already do, you're going to finish the case study you started on Monday. Now, on the last question, okay, so you should have gone through Michael's history. You're reading about a patient describing 2 symptoms, and if you think he's suffering from a genetic disorder, and if you don't know what a genetic disorder is, there should be a link so you can watch it, what a genetic disorder actually implies. Part 2 is when you're actually going to be looking at protein synthesis for a specific gene that codes for a specific protein, hemoglobin. And you may have heard of hemoglobin. It's the protein responsible for transporting oxygen in the blood. So this is sort of the protein synthesis component right here. So normal patients has this code right here for this little strand. And patient Michael has this code. And so you needed to transcribe it to the mRNA and then translate it with a codon chart to form the amino acid sequence. And then just answer these questions right here, 1, 2, and 3 regarding this. When it asks the phenomenon, it's asking you, what is the term that we've used when there's been a change in the sequence of the DNA? It is a one-word answer. I will give you that hint. So that's the protein synthesis. And then we're going to look at see what happens when there is this phenomenon, when there is this change in just one letter sequence. Well, what could happen? And so now we're taking it to looking at the protein structure. And then we're going to take it a little bit bigger from the protein and look at the actual red blood cells that it's in. Now, last question says, use the internet or other resources to develop a claim as to what the disorder of the patient Michael is suffering. Yes, it says disorder. Give evidence. When it says give evidence, you need to use information, especially from all parts, 1, 2, 3, and 4. Okay, the evidence is the data. You don't have to, the evidence, when I say evidence, you're just saying, well, what was the information in that section? And then the reasoning is going to be explaining, well, what that means. Okay, so the data might be that the normal strand here, DNA, and the patient's DNA is different because the patient says GTG and the normal says GAG. That would be the data or the evidence. The reasoning would be, well, what does that mean to your polypeptide or your protein at the end? Okay, and what does that mean for the shape of the protein? So that's kind of the difference between evidence and reasoning. So you can go ahead and use the Oreo format. It's pretty much the same as claim, evidence, reasoning. But I want the claim, okay, or your opinion of what the disorder is that he's suffering, reasoning and evidence in this. Got it? So when you are done there, there's time, some of you go at different, go faster, work at different paces. There are a list of terms that you need to be familiar with. These are all vocabulary terms, like specific to biology, specific to this section, for you to know. So I've included a link for a Quizlet. Okay, so that way you can practice going through them. You can use them just as flashcards. You can use them as a match game to check you, but you can click right on the link there. I've also included extra practice just for checking for understanding. So Quizlet is for vocab, just vocab, what does that word mean? The Quizzies is for checking for understanding. It's more like the concepts. And so I hope I didn't just, yep. So when you go into the Quizzies, it should be a link directly there. I think there's about 16 questions. And then lastly, this is sort of an overview of the central dogma. It's in the same website as our textbook, the text readings I give you, but it's going to kind of give you an overview so you can test your knowledge of looking at a model of the central dogma going from DNA to RNA to protein, and you're going to have to see if you can move the red points to where they belong here and then answer some questions about it. Okay, so hopefully these resources will help you get a better understanding. Friday, we're going to be doing one more protein synthesis mutation lesson. After that, you will start a research project, and I'll start teaching about inheritance, variation in traits, genetics, and evolution. We will also include a do-it-yourself DNA optional lab next week. For this inheritance, teaching about where's that? Now, the protein synthesis mutation lesson, it's going to be pretty similar to the sickle cell one, where you're going to have to transcribe and translate and compare different sequences of DNA and what the resulting proteins are going to be. So it's just more like a reinforcement of what we're doing right now. Okay, so I hope this helps give you a better idea of what's going on. Use the resources. And then finally, don't forget, use videos. So I have to end on this note because, I like the cheesy music. Okay. And on that note, you guys have a good day. Email with questions. If you can't access anything, email me.